Welcome to Chapter 9. This is all about the cell cycle. Let's get started. So we can ask what might seem like a fairly obvious question. Why do cells divide? Here are just some possible answers if we're thinking about single-celled organisms or even multicellular organisms, as we'll see. Just simply reproduction. So like these amoeba here, there was one, now there are two. If we think more in multicellular type organisms like ourselves, growth and development, you start off very small, a single cell, and become multicellular. And then tissue renewal in that multicellular organism. There's turnover of tissues. Tissues are replaced over time. And so all of these things require cell division. We'll focus primarily on the eukaryotes. And again, you've seen this slide previously. Look at this contrast between prokaryotes and eukaryotes. We will touch on how prokaryotes divide, but really there's a lot more going on with eukaryotes, and we'll see how that works when the cells divide. So most cell divisions result in genetically identical daughter cells. So here we have our original parent cell. The DNA is copied very precisely, and then that is split into the two daughter cells as we move along here, and you end up here two identical daughter cells. And so this means these are genetically identical, more or less, than the parent cell. A few maybe nucleotide copying errors are made along the way, but for the most part, these are pretty much identical. So let's look at that genetic material and see how it's packaged and how it works, and start off with a definition here. So you've probably heard this term before, the genome. The genome is all the DNA in a cell. Now you could think, again, if it's a multicellular organism, most of those cells are also going to be genetically identical. So you might just sort of broadly think of an organism's genome as all its DNA, but specifically that's the DNA in one cell and all the genes that are tied with that. So in prokaryotes, that might be this, say, a single chromosome. So prokaryotes typically have a circular chromosome here, like we see here. It's extended out of this cartoon cell to give you a sense of what this is. Whereas eukaryotes tend to have multiple chromosomes. So they have more than one chunk of DNA as part of their genome. And so either prokaryotes are package their DNA in these circular chromosomes, and eukaryotes tend to package them in linear chromosomes and multiples of these. So in eukaryotes, this genetic information is packaged in something called chromatin. What makes up chromatin? All right, so chromatin is a complex of DNA and protein. And so we see here the DNA strands are wrapped around these proteins. These are called histones. The histone plus DNA wrapped around it is called a nucleosome. And then those are kind of stacked together, and then those are stacked together there, and then tightened into coils, and so on. Now, in this cartoon here, you see a condensed chromosome. This might be what you might imagine when you think somebody says chromosome. This might be what you think of. But remember, you really only see that during cell division. So here we see those X-shaped type chromosomes. They're condensed for cell division. The rest of the time, your chromatin is relatively loosely packed because think of it, those genes have to be turned on and off, and all the machinery that transcribes those genes has to be able to get to them. That doesn't work if they're tightly packed like we see during cell division. And so we have these different areas of chromatin in the nucleus here. The dense stuff is called heterochromatin, and the looser, less tightly packed areas are called euchromatin. All right, so eukaryotes have multiple chromosomes, and so it might be helpful to count how many chromosomes different species have. And when we do that, we create something that's called a karyotype. So think, karyote means nucleus, think eukaryote, prokaryote. And so this is a way of visualizing the contents of the nucleus of a certain eukaryotic cell. And so in this image on the right, we see what a karyotype here. So you see pairs of chromosomes, and they're colorized here, so it's helpful to see. And each organism is going to have a characteristic number of chromosomes. So let's take humans, for example, and this is a human karyotype. It is a male or genetic male karyotype. And if we count up these pairs of chromosomes, we have 23 pairs. And if there's 23 pairs, that means we have 46 chromosomes in total. Another term that you'll see soon is this idea of homologous pairs. So think of it like this. You've got really two versions of chromosome number 1, for example, and the genes that are on chromosome number 1 are in the same place on this version as this version, and those variants, I should say, of those particular genes. Let's take eye color, for example. There's a couple of genes for eye color on chromosome 15. The variant on this one over here on the left might be different from the variant on this one on the right, but either way, that actual gene is in the same place on the actual chromosome, just the variants might be different. All right, so if we're looking at non-reproductive cells, these are called somatic cells. Humans would have 46 chromosomes, just like we just talked about. And again, those are two sets of 23. So these somatic cells are considered to be diploid. And so this has to do with that chromosome number. It's two sets of 23, not just 46 totally different chromosomes, it's these two sets. And then when we look at gametes, so the reproductive cells, sperm and eggs, they have half as many chromosomes. So they have 23. So really just one set of that 23 in humans. Again, it's different for different organisms. I'll also say that a different organism might have also 46 chromosomes. It doesn't mean that it's human, right? A chromosome number can be useful as a characteristic, but it doesn't tell you much about, say, relationships. For example, dogs are mammals and we're mammals, but we have a very different number of chromosomes. All right, back to this idea of haploid gametes and cell division, because that's what we're really talking about here. So those typical cell divisions where you have a parent cell replicating, you get identical daughter cells, that's going to go through a process called mitosis. So daughter cells, same amount of DNA as parent cells. But in this other process in cell division called meiosis, we'll get to that in the next chapter, this is how gametes are formed. You're really going through a second round of cell splitting, as we'll see, and you end up with half the amount of genetic material as what you started with in the parent cell. All right, here's a question for you. If somatic cells in elephants have 56 chromosomes, how many chromosomes would you find in a gamete produced by an elephant? All right, yeah, it would just be half that number. So this is the diploid number, 56. The haploid number would be 28 in this case. All right, so let's look back here at chromosomes in a little more detail. So when a cell is preparing to divide, each chromosome gets copied, and then you end up with two sister chromatids. So there's some very specific terminology that we use for this. And so here's an example, a real world, this is an electron micrograph, so you're actually seeing them. So there's sister chromatids here. Before cell division, there's only one of these, and then it's copied, and then you end up with those X-shaped sort of structures that you might think of as chromosomes. But remember, that's only during cell division that they look like this. Another important feature here is the centromere. This is where the chromatids are closely attached, and each one of them has a centromere. And then we'll see how later during cell division, this is going to be a place, the centromere, that we see microtubules attaching and will help pull these two apart into the daughter cells. So here's another way of looking at it just in cartoon form and a couple of other terms to pay attention to. So here are homologous chromosomes. Let's say this is chromosome number 1. And in your diploid cells, you have 2 versions of chromosome number 1. They're just colored red and blue here just for convenience. Each has a centromere. Another important feature here is something called the kinetochore. This will come up again. This is going to be a place where microtubules attach. So prepping for cell division, each chromosome is copied. Now you've got 2, and they've become more densely packaged and form these sort of X-shaped structures like you might think. And again, they're still homologs. Now we just have two copies of each one, but you can see the brackets here. The blue guy is a homolog of the red one over here. And then within those, you have sister chromatids. So the blue one and the blue one, those are sister. Red one and the red one, those are sister. Okay, so in a cell in G2 of the cell division cycle, it contains 20 chromatids. So how many centromeres are present? So I'll clarify, in G2, the DNA has replicated at this point. And so how many centromeres are present? All right, so there are 20. It's the same as the number of chromatids. Now, this takes into account a bunch of different things. Understanding what a chromatid is, that's each version of those chromosomes, and each one of them has a centromere. So understanding that gets you quickly to that answer of 20. So let's look now at what happens during cell division and chromosome replication. We'll just focus on this organism, hypothetical organism that just has this single chromosome just for convenience. And we can think about how much DNA is present at each step as well. So after leading into cell division, the DNA will be replicated and then it will condense, and then you get your condensed sister chromatids here like we've seen before. And really we have twice the amount of DNA as we started with, right? And this is not just DNA isn't just going to keep doubling because, well, what happens next? Those 2 sister chromatids will be separated into the daughter cells. So each daughter cell is going to have the same amount of DNA as the parent cell, and each has that 1 chromosome in this case. All right, so you've learned a little bit about chromosomal duplication, some of the anatomy of those chromosomes that will be important as we get into some later stages here. Let's look more broadly now at the whole cell cycle. I'll point out right away, because this is often a misconception, because we're talking about mitosis in this chapter, that sometimes students will think, well, the whole cell cycle equals mitosis. No, mitosis is just a part of the cell cycle. It's called M phase. And we'll actually see later with meiosis where you really sub in meiosis here in place of mitosis. We'll get to that in the next chapter. But then the rest of the cell cycle is called interphase. So together that makes up the entire cell cycle. And we're going to start by looking at interphase. So this is most of the cell cycle if you think of in relative to time. So there's 3 phases. The G1 or gap phase, and this is a time of cell growth. The S phase or synthesis phase, synthesis of what? DNA. So this is really important, right? We've just been talking about chromosomes being copied. And when does that happen? It happens during S phase. And again, this is not in mitosis yet. We're not even there. This is still part of interphase. So we get from 1 chromosome, we create those 2 sister chromatids. Then we enter into gap 2 phase or G2 phase. This is where we see those chromosomes really condensing. And like in G1, it's kind of a time of cell growth. The cell is really preparing for that next phase, which will be mitosis. So G2, as we get close to mitosis here, those chromosomes are becoming condensed. And another important thing happens. The centrosome This is that microtubule organizing center. Think back to the various cell structures that we talked about in an earlier chapter. These replicate and each moves to a different pole on the cell. So here we see in this cartoon up here, there's 2 of these centrosomes now, and they're going to kind of move in opposite directions. And these are going to be anchor points, as we'll see later. And I will point out now that plants don't have these organizing centers. They basically use other structures and use the cell wall of the plant to anchor it. But the animal cell needs these anchor points. So now we're moving into the first phases of mitosis as those chromosomes become condensed in G2. And just a reminder that condensation of the chromosomes is really important. This is going to prevent them from getting all tangled together as they need to be separated into those 2 daughter cells. And so let's look at how they're going to be separated in a minute, and focusing first on these structures here shown in green. This is called the mitotic spindle. So this spindle apparatus is made out of microtubules. Think back to cytoskeleton components. And these are really important for controlling where those replicated chromosomes go and then ultimately separate into those daughter cells. Those centrosomes are those anchor points. So as we're seeing MTOC, microtubule organizing center here and here, and then those microtubules will attach to the sister chromatids, and this is going to help them separate, as we'll see shortly. The attachment of those microtubules occurs specifically at something called the kinetochore. You saw this before when we were looking at the anatomy of sister chromatids. So the centromere is here, and attached to it is this kinetochore structure, and here is what the microtubules actually attach to. And those spindle microtubules that attach, we have a specific name for them. They're the kinetochore microtubules because they attach to those kinetochore. And that happens, as we'll see, during prometaphase. So here's the really zoomed out view of what the cell cycle looks like. The cell grows, DNA replicates, those chromosomes are then split into 2 daughter cells, and that is this last bit is the process of mitosis, and we'll see how that works. Mitosis is split into 5 phases, and they're listed here. The last phase that somewhat overlaps with the later stages of mitosis, but typically it's given its own category, is cytokinesis. That's ultimately where the cells split into 2. From 1 cell, you've then got 2. The stages of mitosis, fairly easy to keep track of if you know some of these prefixes. So meta means middle. So that's, if you got 5 things, that's the middle phase. Telo means end. Pro means before. And then prometaphase would be in between these 2. And then anaphase is the last one to fill in at step 4 here. So let's see what happens at each of these stages. During prophase, our condensed chromosomes become very visible. So that started happening in G2. And here we see in prophase, in this stained preparation, very clearly chromosomes. The nuclear envelope that surrounds the nucleus, that begins to break down. That has, in this case, has to disappear. That spindle apparatus is starting to form and extend those microtubules out. Prometaphase is next. So what's happening here? Here's where we see those kinetochore microtubules actually attaching to those sister chromatids. At this point, the nuclear envelope is gone. It's been dissolved. And here's an actual image of what prometaphase might look like. You see the microtubules staining red here, the blue chromosomes, and they're still kind of all jumbled up a little bit. As we'll see, that's going to distinguish the next stage. Here, if we look more closely at our sister chromatids during prometaphase, we see those kinetochore microtubules attached at the kinetochore. So next up is metaphase, or middle phase. At this point, remember, our microtubules are attached to our chromosomes. This, through a bit of pushing and pulling of these microtubules, our sister chromatids are going to line up in the middle of the cell here. And this alignment is important because ultimately the cell needs to get one of each version of these sister chromatids into those daughter cells. At this point, those sister chromatids are held together by something called cohesins, these proteins called cohesins. Think cohesive, they're stuck together. And a real image of what this looks like, a little bit different than prometaphase. All the chromosomes are lined up in the middle of this image, and the red, again, are the microtubules attached. So here's what this looks like in a little more of a 3D imagining of what this looks like. Here's our centrosomes, microtubules radiating out from it, and this imaginary plane in three-dimensional space is called the metaphase plate. And so that's where the chromosomes are lined up at this point. Again, think of taking a sphere and slicing it in half. That's the same idea of this metaphase plate. That would be like an imaginary slice through the middle of this sphere. All right, before we go on, here's a question for you. There is a protein known as colchicine that can block the formation of that mitotic spindle that we've been talking about. So what would happen if you add colchicine to cells in G2 phase? What's going to happen? All right, so here were your choices. You might have got them in a different order. The answer is D here, that chromosomes, they will condense because you don't need the spindle for that, but you're not going to get the attachment during prometaphase, and then you won't get the alignment like we just saw at the metaphase plate during metaphase. Why not these other ones? Well, the cells wouldn't immediately die. The cells would be able to begin mitosis, but they won't get very far in it. And the nuclear membrane certainly, yes, would break down, but no, the chromosomes would still condense. So that rules out this one. All right, so everything's working normally here in our example. We're into anaphase now, so this is the fourth phase. Now remember, those cohesin proteins, those were holding our sister chromatids together. At this point, if everything's working correctly, in comes this enzyme called separase. It's kind of a convenient name. That will cut those cohesins and allow the sister chromatids to separate. So they will separate, and then those microtubules will move them toward the poles. Now, an interesting question here is, are the microtubules shortening right next to the chromosome, or are they shortening down by the centrosome, or is it a combination of both? And so this is a cool little experiment described in your textbook where these researchers used a molecular mark on the microtubules to see where they were being shortened. And so here would be just jumping to the results here. What they saw was the shortening tended to occur out by where the microtubules are attached to the chromosome. So what that means is these motor proteins at the kinetochore are moving the chromosomes along, and that tubulin is depolymerizing as it happens, and that's how these are getting shorter. Now, this is a very cool experiment, and we know a little bit more. There's an added nuance here. Although most of that shortening is occurring right here on this end at where it's attached to the chromosome, the microtubules do also shorten to some extent out at the centrosome. All right, so there's a few other things going on here with the mitotic spindle at anaphase. So the sister chromatids separate, absolutely. Those kinetochore microtubules are shortening, so we're pulling the sister chromatids away from one another, as we see down in this image here. But another thing that's kind of cool is these non-kinetochore microtubules, they help really stretch that cell out, and not only stretch the cell out, but pull those poles apart. So how is that working? Well, there's little motor proteins attached to these, and they're just working against one another. So they're pushing away from one another. So that's extending the distance between these two poles. And that's going to lead us right into telophase. So here we see anaphase, that cell's elongating. Here are those overlapping non-kinetochore microtubules. what's happening during telophase. You start to see the nuclear envelope begin to reform. That's shown here in this kind of cloudiness here. The chromosomes start to uncoil, and they're basically where they need to be on opposite ends of this expanding cell. So telophase leads right into the last stage here, which is cytokinesis. And this is where we see the cell split from 1 into 2 identical daughter cells. In animal cells, you first see this cleavage furrow form, so this indent. There's a bunch of proteins that kind of squeeze between the two poles, almost where that metaphase plate was. You can imagine that. And it's going to cause those cells to basically pinch off and produce 2 daughter cells. Now, in plant cells, this kind of thing doesn't happen. So after the DNA replicates and undergoes mitosis through telophase, a new cell wall will start to form between those 2 daughter nuclei. This is derived from the Golgi apparatus. So these vesicles form down the middle here from the Golgi, and they coalesce and start to form what's called the cell plate. And then that becomes a new cell wall. And here we have now 2 daughter cells separated by this cell wall. So a little bit different in plants and animals, but the gist of it is the same. You started with 1 cell, now you have 2. Now you might want to take a look at this plant cell division image here and see if you can identify some of the different stages. This one down here toward the bottom right, I think this is a good example of telophase, almost leading into cytokinesis. I can see what's probably the forming of a cell plate here. If we wanted to find anaphase, that's kind of an easy one. Here's this cell's definitely in anaphase. The sister chromatids are being separated. Pause it here. See if you can find some of those other stages. All right. Thinking about cytokinesis. What's going to happen if a eukaryotic cell undergoes mitosis but does not undergo cytokinesis? All right. Yeah, so you'll end up with a cell that has 2 nuclei that are identical to one another. They're daughter nuclei, but the cell hasn't split in 2, so you end up with this cell with 2 nuclei. All right, so what happens in prokaryotes? It's a little bit different, right? Think back to earlier in this presentation. Prokaryotes typically have a single circular chromosome. They don't have a nucleus, so it's a little less complicated there. So how prokaryotes divide is much simpler, really. The DNA has to be replicated, so that circular chromosome is copied. Now you got 2 of them. Where it starts at what's called the origin of replication. We'll see how this works in DNA replication in a later chapter. We had 1 chromosome, now we have 2. And then these are just separated into 2 daughter cells. We see this like cytokinesis-like event, but this is not called cytokinesis here. It's called binary fission. So there's proteins that help form a barrier between these 2 daughter cells as they elongate. And we had 1 cell, now we have 2. So that's binary fission, and this is what prokaryotes do. So let's put cell division into a broader evolutionary context now, and thinking about the evolution of mitosis. Well, binary fission probably evolved first, and then mitosis evolved from binary fission. And why do we think that is because we know from the fossil record that prokaryotes evolved first. So we can look at this timeline of life on Earth here, 3.5 billion years ago, the oldest fossils. These are prokaryotes. We start to see oxygen in the atmosphere 2.4 billion years ago. And then the evolution of eukaryotes somewhere between, depending on who you ask, 2.1 to 1.6 billion years ago. And so eukaryotes evolved later. And so if we actually look at some other eukaryotes, we see hints of binary fission in how they undergo cell division. And so these are a couple of examples from your textbook. So if we look at these dinoflagellates as an example here, something interesting that happens with these is the nuclear envelope doesn't actually dissolve like we've seen already. The microtubules actually pass into the nuclear envelope here. So it's almost like the nucleus itself is almost undergoing something like binary fission, which is really interesting. With, of course, those microtubules passing through. So it's a little bit intermediate. Diatoms and yeast, this is where we see the nucleus dividing, again, sort of like binary fission. The nuclear envelope doesn't dissolve. It stays completely intact. The chromosomes are separated into the daughter nuclei. And so this itself looks a bit more like binary fission. Again, the differences would be, well, there's microtubules here. So it is certainly different, but these are reminiscent of binary fission. All right. So in the next part, we'll talk about control of the cell cycle. See you then.